19th century, the active ingredient explaining the biological activity for anti-malarial biological activity was assigned to quinine. And this was historically one of the first, maybe the first drug which proved to be effective against malaria. So, if you look at the historical timeline, and this is borrowed from a review, starting from quinine here in blue, all the way to artemisinin, which also comes from a natural product drug, which here in this case from traditional Chinese medicine, as you know. You have a series of drugs which have been developed, so each time a drug was introduced is shown in blue. But very soon after, and it's indicated in red in this diagram, you had a resistance to the drug which was happening. And as a result, it's an ongoing battle over the centuries to come with new compounds showing promising antimalarial activity. Currently, the standard of treatment is a combination to fight resistance, is a combination of artemisinin and another longer half-time drug, which can be primaquine or other drugs. So this means again that there is a need to find new targets and new compounds to keep the hope that we can still cure malaria, even if we don't have vaccines have been deployed, but they are not so effective at this stage. So we still need a big effort to make a big effort in terms of drug development. So one of the I would say efforts, joint efforts, which has been happening in the last decade or so, was to assemble libraries of compounds which showed in phenotypic assays against the whole malaria parasite, in phenotypic assays, which showed promising activities. And one such example was provided by the Medicine for Malaria Venture. This is their website. And they collected about 400 drug-like compounds which all showed promising activities in terms of killing the parasite in a cell-based assay. But in many cases, the mode of action of these compounds is not clear. And in order to do medicinal chemistry, obviously, and to improve the potency and selectivity of these compounds, it would be useful to have a method to deconvolute, to understand how, the mode of action of these drugs. And this would eventually lead to improving their potency. So in terms of another drug I'm going to talk about in terms of mechanism of action, this is MK4815, MK for Merck. So it was developed by Merck using their compound library, and is also identified phenotypically. It's an interesting drug because it's very cheap to synthesize. It's effective in a Plasmodium berghei animal mouse model against malaria, and it's also active against the prototype 3D7, but also against several field isolates which are resistant against many drugs. But again, here for MK4815, the mode of action was not understood so well. So what I'm trying to convey to you today, this morning, is how can we figure out the mechanism of action of some of these compounds, the old one, like the quinine, the quinoline compounds, but also of new drug candidates like MK4815 from the Merck compound. So why do we want to do that? Again, just to emphasize the point, because a better knowledge of their targets would help us to improve the selectivity potency, and so we need to do target deconvolution. So one way it was done, it's been done, is using a combination of in vitro selection of resistant parasites, followed by whole genome sequences sequencing to identify mutations that appear to impart resistance to the parasite that was grown in the presence of the drug, figuring out that there has to be a connection between these two. Another method which is used is the use of clickable probes. So you modify the drug, and then you can fish out the putative targets in the ocean of the possible targets, let's say in the cell lysate. So another method that I'm going to talk about today is mass spectrometry cellular thermal shift assay, the long name. So MS-CETSA, or CETSA for short, is a proteomics-based method that was initially designed to deconvolute drugs targeting cancer. And it was repurposed by Professor Bosdek in our university in Singapore to try to understand the mode of action of drugs, anti-malarial drugs. So how does it work, this CETSA method? It works on the following principle, that once a drug binds to a protein, this protein becomes more resistant to thermal denaturation and precipitation. So it tends to, its melting temperature is shifted towards a higher value compared to the case when there is no drug. So binding induces stabilization, a thermostabilization of the drug. So if you look into details, into a little bit more details, but there are reviews, I'm not going to go into too many, but you have two panels. So the one on the left is the, what I just said, the melting curve. So what you do is you use a single drug concentration and you increase the temperature. So in the absence of drug, you have the black curve, meaning that you have the proteins which gets precipitate and denature at a certain melting temperature. But in the presence of the drug, a number of putative targets become stabilized, and they are more resistant, more resilient to thermal denaturation. And then you go from the black curve to the red curve, which is shifted to the right, meaning that the melting temperature is higher. So you can measure the area which is in between these two curves, and it gives you the readout, the significance about the destabilizing, thermostabilizing effect imparted by the addition of this drug. So you come up with a list of compounds. You have another implementation of the method, which is described on the right panel. So here, instead of using a variety of temperature, you use a single temperature, and then you increase the concentration of the drug. Here on the x-axis, you increase. So at low drug concentration, compared to the black control, there is no variation because the drug is in a too low concentration to be effective and to impart, to stabilize the target. But when you increase the concentration of the drug, then you have a number of proteins which become thermostabilized, and then you depart from the baseline. And this is again, you have an area of the curve which gives you how reliable the signal is. So what we did is we selected a number of drugs we wanted to understand the mode of action, and we performed this MS-CETSA experiments. So here I'm showing a few compounds that we passed through this this CETSA method, and it came up with list with this significance value that I showed you in the previous slides, and we could come out with a number of putative targets for chloroquine, for mefloquine, and for the Merck compound 4815. And so this we have a number of publications which describe this in details, but one result and one protein that came often was the falsilysine, which was stabilized by many of both the quinoline compound, but also of drug candidates like MK4815. So we were interested in falsilysine. Why? Because falsilysine plays a key role in hemoglobin digestion. So during the life cycle of the parasite, to provide for amino acids the parasite scavenges amino acids from the hemoglobin within the red blood cell, and this provides this amino acid for its own metabolism. So if you look at proteins whose expression is upregulated and they are displayed in red, so in the presence of this drug, either chloroquine or MK4815, you have an increase of the expression of the hemoglobin digestive enzymes when the parasite is incubated with the quinine, chloroquine, or MK4815. If you look at the chloroquine-resistant DD2 strain, you don't have this effect. So this suggested to us that these compounds, also chemically unrelated, could target falsilysine. So we wanted to understand a little bit more in this. So just as a reminder in terms of the activity of this, so it's a zinc metalloprotease, which is involved in degradation. So it's part of a cascade, a proteolytic cascade, which is displayed here. And it's also part probably of a multimolecular complex in the food vacuole, which is one of the compartment, low pH compartment in the parasite, where you have processing of, so you go from the whole alpha-beta hemoglobin chain to smaller polypeptides, and eventually to single amino acids using this cascade. And falsilysine is thought to digest peptides about 10 to 20 amino acid long into smaller fragments that can be further digested. So we wanted to understand how, so several questions. How does falsilysine recognizes its hemoglobin substrate? That's one. And also, how could drugs like chloroquine or MK4815 were able to inhibit the activity of falsilysine? So to do that, we have a nice platform of protein expression and purification. So we did this, so it's 139 kilodalton protein that we expressed. It's E. coli expression, so it's not too difficult. And this is the purification we come up with. And the first thing we did, based on the CETSA results, we wanted to confirm using the recombinant falsilysine protein. We confirmed using ITC, isothermal calorimetry, binding of these compounds to the falsilysine, because it's always good, of course, to check the results, because from going from a cell lysate or whole cell assay to individual recombinant proteins. So on here you have on the various panels, from the left you have the binding of MK4815, of chloroquine, mefloquine, and a negative control, which is actually artemisinin derivative. So not everything binds to falsilysine. So here, I'm not sure you can read, but you have the thermodynamic parameters for each of these compounds. It turns out the one that binds best is the Merck compound, the MK4815. So we kept going along the same line, and having checked the binding of these compounds to falsilysine, we checked whether these compounds were able to inhibit its enzyme activity, its protease activity. So to do that, we used a peptide substrate, which was fluorescently labeled with dapsil and edans on this N and C-terminal N, and this is the cleavage site. So, so the enzyme, the proteolytic inhibition assay was performed at 2 pH, at neutral pH, and also at the pH, acidic pH, which is found in the vacuole, in the food vacuole, just to mimic these biological conditions. So here again, the best inhibitor in terms of the best compound which inhibited with the lowest IC50 the activity of the falsilysine was the Merck compound 4815. So the question then was, how does So it seems, conclusion of this part, it seems now we have identified a putative target of falsilysine. So for a putative target of MK4815, and eventually also of chloroquine, which binds falsilysine with a low micromolar affinity and also inhibits its catalytic activity. So next, we determine X-ray crystallographic structures. So these are experimental structures. It's not a model with the electron density. So we have the complex here of the one quinoline compound shown here. The quinoline compound in a hydrophobic pocket of the falsilysine. Likewise, we determine the Merck compound, which is here, bound to the falsilysine. And it turns out they bind to the same pocket, and you will see where it is in the structure in a couple of slides. So this is a summary of the crystallographic work, where we have chloroquine, so two quinoline compounds bind to this inhibitory pocket of falsilysine, and the Merck compound bind to the pocket. So just here for those who like crystallography, we took, as you know, chloroquine, you have halogen chloride group, and we use the anomalous signal from the chloride atom to make sure of check the orientation of this compound to make sure it was properly built in the electron density map. So you see this is pink. So here actually you have halogen pi interaction, which imparts quite a lot of specificity to this interaction, even though the affinity is not that big. So where does it bind? Where do these compounds bind? In the falsilysine structure. So this is shown schematically in here. So you have this surface representation of falsilysine, and you have the active site, which is somewhere here, where the zinc binds. And at a distance of about 20 angstrom away, you have a pocket where the compounds bind. And the question was how, by binding distantly from the peptide substrate, how does this compound are able to inhibit the activity, the proteolytic activity of this enzyme. So we did, on the way, we did a little bit of, I would say, site-directed mutagenesis to confirm that what we observed was true. And we looked at the Merck compound, the most potent compound, and we observed that residue glutamate 530 made a hydrogen bond with a hydroxyl group from the Merck compound. So we say, if the structure is correct, if we mutate this glutamate residue, which gives a hydrogen bond to the compound, into an alanine, then we would disrupt this hydrogen bond, won't be there anymore. And that's what we did. And we measured again for this single mutant of falsilysine. We saw a decrease in the binding affinity, about 40-fold, and also an increase in the IC50, the enzymatic inhibition activity. So this gave us confidence about the reliability of the structure. And also, to some extent, it allowed us to measure the strength, the energy given imparted to the interaction by a single hydrogen bond. So the next question we wanted to address is this one, is, by the way, how does the falsilysine, which is a very important enzyme in the hemoglobin degradation cascade, and by the way, it is, if you knock it down, the parasite cannot survive, so it's an essential enzyme for the parasite life cycle. So that's also why we did all these studies, because it is an important enzyme and probably a good drug target. So one question which has not been addressed so far is, how does falsilysine recognizes its peptide substrate? So we use crystallography again to address this problem. And initially we used the wild type enzyme, but then whenever we soaked the peptides got cleaved, and we couldn't see anything bound in the structure. So what we did is we generated, we look into the active site, and we mutated the glutamic acid 132 into a glutamine, and this disrupts the proteolytic activity of the falsilysine. And then we co-crystallize the enzyme, this mutant enzyme, with two hemoglobin substrate, one of 15 residues, one of 14 residues, which come one from the alpha chain and one from the beta chain. So I will call them alpha peptide and beta peptide in the following. So we got high resolution structure. And so on the left side is the structure that we got of falsilysine bound to the alpha peptide, and on the right with the beta peptide. So just as an aside here, you also have the first view, I mean a more detailed view of how falsilysine looks like. So it's a big enzyme, so it's about 1,300 amino acids. So you have an N-terminal domain in blue. The N-terminal domain is about 630 amino acids, and you have a linker region, which is here, which connects the N-terminal domain to the green domain, the C-terminal, also about 630 amino acids, in a flexible way. So in other words, the two domains can move relatively to each other. And why is it important functionally? Because you need to give access, the enzyme need to give access to the substrate into the catalytic chamber. So imagine the peptide has to diffuse in, then it gets cleaved, and then it gets released. So here we have a view of how the enzyme recognizes its hemoglobin substrate, so for the first time. And you have similar conformations for the two alpha and beta peptides. And note that if you look at the corresponding structure in hemoglobin, is not the same. It adopts an alpha helical structure. So it means that the falsilysine recognizes already a denatured form of hemoglobin, which makes sense if you look at the cascade. Falsilysine cannot proteolyze the whole enzyme. It has to be already digested into smaller pieces. So if you zoom in, you have a more detailed view of how the enzyme recognizes the peptide substrate. And this is shown here on the left for the alpha peptide, on the right for the beta peptide. And one, I'm not going into too many details, but one, the most conspicuous feature of this enzyme peptide interaction is the formation of an antiparallel beta strand. So you have a strand in blue of the enzyme, which makes an antiparallel beta strand interaction with the peptide here, the hemoglobin alpha peptide substrate. So this is a trick, I would say, used by the enzyme to, because as you know, in a beta strand, the side chains come either towards you or away from you. And it means that for many sites, there is no specificity of recognition, because the recognition comes from the main chain, hydrogen bonds, which is largely sequence independent. So that's a trick used by the enzyme, so that you can recognize a variety of peptides and cleave several different peptides. So here you have, in blue, you have the zinc, which allows this peptide bond to be cleaved, indicated by an arrow. So if you compare now that we have an experimental structure of the peptide bound in falsilysine, we also had another structure of the MK bound to the structure, as I showed you earlier. So how does this compare? So you see actually that there is quite a distance between the peptide and the inhibitor binding site. So as a conclusion of this, we can say that, and this fits the enzyme kinetics measurement, the Merck compound is unlikely to directly compete with the peptide binding. So how does it work? And this is, I just go quickly, this is the Lineweaver-Burke measurements where we found an uncompetitive mode of binding. So the structural data match here the enzymatic mode of inhibition data. But still, we don't have a complete answer. So at this stage, we have found how falcylysine binds its hemoglobin peptide substrate. We have found how it binds the compound, but we don't really have an explanation of how the Merck compound inhibits falcylysine. So we have to come back to, so falcylysine, we have snapshots of it, but it's a dynamic enzyme. So how does it work? I showed you there is two lobes, two parts, the blue and the N-terminal blue and the N-terminal green, C-terminal green. So it has to cycle between various conformations. One of these conformations has to be open. So the two domains, the linker allows this opening. And this allows the substrate, the peptide substrate, to diffuse into the catalytic chamber, which is very large, and sit, as we have seen, inside the chamber. So once it's diffused in, the enzyme closes up, and then the catalytic site is formed, is put in place, and then the zinc metalloprotease is active. It cleaves this up, it cleaves the substrate, and then the enzyme has to open again, so the product diffuses out. But we don't know the details of how this works, and what we have in crystallography is only the closed form, this form there on the bottom right. We only have the closed form, which so far we don't have any view of the open form. How does the enzyme look when it's open? So to do this, we used another structural biology technique, which is available at NISB, which is the cryo-electron microscopy using the Titan cryos. And what we did is this following experiments. We first set on the grid the falcylysine without any compound. This is what we obtained on the left. And on the right experiment, we pre-incubated the Merck compound with the enzyme. And we did 2D reconstruction, and we just counted how many particles are in which conformation. So it turns out that while in crystallography all the particles that crystallize are in the closed conformation, when you use this technique, more solution-like technique, in the absence of compound, the enzyme is mostly in its open form. It sits open. You have about 2/3 of the particle in the open state and 1/3 in the closed state. A bit like the one we observe in crystallography. If you incubate the enzyme with the Merck compound, then it shifts. You have 1/3 which is in the open state, and you have 2/3 which switches to the closed state. So how do these two states compare? And this is shown at the bottom here. So at the top, so these are the N and C terminal, the two lobes. And when it opens up, is a bit, you have a movement of about 25 degrees, and the two domains separate with each other. So another point here is that we could also, using cryo-electron microscopy, we could see that only the closed form of the Merck compound could accommodate the falcylysine could accommodate the Merck compound, but not the open state. And you can see here you have a too close interaction between this, the drug and the protein. So, so just to summarize this movement, so if you superimpose, for instance, the N-terminal domain, then you see you have a hinge motion going from the red to the green. So the open would be like this, the red, and the closed form is denoted C here. And you have a hinge helix, which doesn't move much during this movement. This is why we call it hinge, which is in the linker region. and allows the protein to perform this opening movement, again needed for its biological activity. So now I'm going to show you this. Yeah. So this is the fitting in the electron density map in the closed form. This is the opening of the falsi lysine to accommodate the substrate in the open form. So just to finish on this part of the story and conclude how does the Merck compound inhibit falsi lysine, you had this hinge helix, which is important to allow the opening, like a door gets open. And Merck actually comes very close to it. This pocket, binding pocket, is very close to the hinge helix. And in presence of this Merck compound, the dynamic of the enzyme is impaired such that there is no possibility of going into the catalytic cycle. So this, yeah, please. So here I just, in this small movie, I showed you the various conformations, the open, and the closed conformation were identified in cryo-electron microscopy and in crystallography. So that's, I would say, one key conclusion. And here you have the binding of the peptide in the catalytic pocket. So conclusion of this part is the following. How does the Merck compound inhibit falsi lysine? It doesn't compete directly with the peptide. What it does, it takes away, it sequesters away the enzyme from the normal catalytic cycles by preventing the close to open transition. So somehow you have a number of enzymes which are not able to enter the open-close transition, which is needed for during the catalytic cycle. So the second story, which will be not as long, but just to conclude, is another example of the power of the method, is how does one of the MMV compound work? There again, I told you there are more than 400 compounds which have been collected, and which shows the 848 compound, which is also, I would say, promising in terms of anti-malarial activity. And we identify, using CETSA as well, a number of putative targets. And there we found that one enzyme, which is the purine nucleoside phosphorylase, PNP, was quite stabilized, thermostabilized by addition of the 848 compound. And this was done either in an intact cell CETSA assay or using a lysate cell assay. So here I switch. I have moved from my falsi lysine story to the purine nucleoside phosphorylase. And this is an overview of the metabolism, the purine metabolism in the malaria parasite. So you have a number of enzymes which are involved. And the purine nucleoside phosphorylase is here using in this catalytic step, using this hypoxanthine as a substrate here. So I think it's a bit clearer probably in this slide. So what it does is convert, it takes the inosine and the phosphate as a substrate, and then come with the hypoxanthine as a product and ribose 3' phosphate, which are important then for purine nucleoside metabolism. So this is also interesting drug target like falsi lysine is. So what we did is here a thermal shift assay. So CETSA has identified PNP as a putative target, And what we did is we took the 848 compound and we do the simple thermal shift assay, non-expensive assay. And you see that in presence of increasing concentration of drugs, you have a very large shift, about 20 degrees shift in thermal stability of the recombinant PNP enzyme. Likewise, we did the ITC measurement, which is shown here, and we found that 848 selectively binds to PNP, which is shown on the left. So the question is, and in respect to, there are some groups who work on PNP, both from the human, it's a cancer target, and imucilin, as you know, imucilin is a known human inhibitor of human PNP. So we wanted to know the selectivity of 848, whether it can bind the human, and it does not. As you see on the right side, when you do a binding assay with human PNP, there is no binding, so it selectively binds to the plasmodium enzyme. This is unlike immunosilin. So likewise, we did a number of enzyme assay. So I'm not going into details, but here we found a competitive mode of inhibition of 848 with respect to the inosine substrate. And what we also found is specific inhibition of the parasite enzyme, while the human enzyme is not inhibited, as you could say from the binding assay. So here I show you so this is the plasmodium enzyme. So we determine the structure with X-ray crystallography. And so it's a butterfly-like structure with 3 dimers. So you have so it's a hexamer. So you have 6 monomers, but they are grouped 2 by 2. So one monomer. So this in blue, you have the 848 compound, which this is a phosphate group, and this 848 sits here in the active site cavity, which is maintained in the cavity by a number of hydrophobic interactions, but also by a network of water molecules. So you have 6 molecules bound, 1 per hexamer. So this morning during the introduction, there was a talk about synergy. So we wanted to test whether there was antagonism or synergistic effect with this imucilin I just mentioned, because imucilin also binds to the active site of PNP. So what we did is we did an isobologram assay where we used jointly the 848 and x-axis and either imucilin or quinine in the vertical axis. And you can see that there is an antagonistic effect with 848, either with imucilin or quinine. So this suggests that these 3 compounds share a common binding site and some common mode of action in the PNP binding site. So this was, I would say, a cell-based confirmation of a mode of action. So now I'm going to reach the conclusions using the 2 examples I've tried to explain to you this morning, one on falsilysine, one on purine nucleoside phosphorylase. So the first one is the CETSA method is a tool that allows the deconvolution of drug targets which have been identified. So you come up with a list of compounds, either using cell-based or lysate assay. And this is only one step, CETSA, but it's a useful step. It gives you hints and possibilities, but that needs to be validated. Now, how do you validate them? You need a thorough, as I tried to show you, biophysical, using thermal shift assay, using ITC, using enzyme assay, and structural characterization that we used here, experimental structure determination using X-ray crystallography and cryo-electron microscopy. So one conclusion of this work, and also the other work I haven't shown, but some of this work is in press in cell chemical biology, and I mean, you can look more details into this, but it seems that the paradigm, one drug hits one target, is not always valid. I mean, in many cases, we have found that one drug can engage several different targets. So this is interesting because maybe maybe we should think a bit differently, that the mode of action is not as simple as sometimes one may think, and there is multiple multi-pronged effects and synergistic effect of one drug engages several targets. Conversely, there are some proteins like falcilizine which are able to engage many drugs. I showed you falcilizine binds the Merck compound and also the quinoline and also other candidate drugs. So for falcilizine, we identified a druggable pocket. So this is interesting in terms of medicinal chemistry. So now we have the structures available. We can do more medicinal chemistry to improve the potency of these drugs. So one difficulty we had is to perform genetic studies to try to elicit resistance against the drug, to confirm the validity of our approach in terms of the targeting. And this comes to the confounding factor, one drug, one target. So these are definitely a sticky point, the genetic studies, and we are working on it. So for the PNP work, we have found that quinine, the chloroquine compound, quinine, the quinoline compound, quinine, mefloquine, and also 848 bind competitively to the PNP active site. And since PNP is key in the purine salvage pathway, we now have a sound basis to develop, I would say, analogs, compound analogs that would be more potent to inhibit PNP activity. And since PNP is very important in the purine salvage pathway, we might have some interesting leads there. So I'd like to finish the work by acknowledging the people who participated. So Tianjin, Lin, and Zara Chung, they are PhD students, were very instrumental, and they worked by Abbas, who is a research fellow. And I'd like to thank the people in ZB, Zbigniew Bostek lab, Jerzy and Gren, who have been very important for the CETSA work. I'd like to thank the collaboration supported by National Research Foundation with Peter Preiser, Bosdek, and Liu Chanfa, and the support in the grants by the Singapore MIT Alliance for Research Technology, the NTU, and the Ministry of Education. And I'd like to thank you very much for your time and attention. Thank you.
Thank you, Professor Julien. Now it's open for the discussion. I mean, one or two questions, can be asked. Okay, yeah. Please tell your name first and from which organization you are. And you can have the questions. Hello.
Can you hear me, Julien?
Yeah.
Hi, here. Over there, here. Thank you. My name is Binay Panda. I'm from Jawaharlal Nehru University, Delhi. I want to be a little provocative in my question with not an intention to offend people in the group, but to elicit a response from you which is more futuristic. What you described is a very old-fashioned, classical, small molecule drug development. Of course, with biophysical, biochemical, X-ray crystallography, and cryo-electron microscopy data. Now we know that this has failed in many fields. Now I want to ask you And you mentioned about the side-directed mutagenesis approach of screening. How are we in the field in general, and malaria in particular, of using some of the deep learning tools of screening small molecules to start with, but not ending there, because instead of spending money, time and efforts doing side-directed mutagenesis, you can actually understand a lot of that by doing structural analysis, because right now people even have, as you know well, included the AlphaFold, RosettaFold data with both cryo-EM density and Excel crystallography to find more. So why visit something which we know really hasn't worked well, and how does the future hold?
Yeah, it's a very valid point. Yeah, I carefully avoided AlphaFold, but I'm very aware of AlphaFold, and we use AlphaFold as well every day. I think it's become a very useful tool. And I agree to some extent, maybe just 2 years from now, this talk will look like even more ancient. Yeah. To the point that maybe AI, machine learning technology will have evolved to the point that there won't be anymore the need to use experimental method of cryo-electron microscopy, X-ray crystallography. And it's true, it's something that you're right to point it out. Yeah. I think today, as we speak, first of all, the technology has evolved in AI. It also has evolved a lot in cryo-EM and structure, to the point that the effort needed to get these structures is counted in days or weeks. So I would say, in terms of validation, it still gives a very useful anchoring point of future experiments, be it using AI or other modeling experiments. So I wouldn't say this, I wouldn't phrase this as antagonistic. I mean, the structure which are deposited are publicly available, anybody can use it, computational scientists should use it. They are there. They can be used to augment, expand the compounds, modify the compounds, and we are doing it. So I don't see it as a contradiction. I see it is one piece in the puzzle. Yeah. For the single mutation, it's. I mean, you can argue, yeah, whether there is a need. I mean, this experiment is already 3 years old, so of course I showed it because yeah, it was done. But yeah, maybe now you can computationally model it, even though the value of the delta G, I'm not sure you can. I don't think you can get it computationally accurately as you do in isothermal calorimetry. So you really get a very accurate value of the enthalpy, entropy, and things. So yes, you're right, but at the same time, I think it's. I don't see it as antagonistic. So the next stage, yeah, is to, and we are doing it, is to grow the compounds. We are other analogs and do this, and we do it also computationally. I didn't present any computational work, but yeah, we are not blind to computation, and we use it a lot. Yeah, but it's a very fair point. And as you say, maybe in 2 years, yeah, the state of AI will be such that you can design a drug automatically. But I mean, this is the purpose of this meeting, I guess, also to discuss the future. Yeah.
Thank you.
Any other question? Yeah, please introduce yourself first. And
inhibitor is, you mentioned functionalizing. So is that has any off target? You know, it can be inhibit to human enzyme because OTGs are well aware in the human. So what is the possibility?
Yeah. So for the fancy lysine inhibition, I would say is a metalloprotease. You have a family of enzymes which are found in various organisms, but there is very little homology. So the likelihood that you do a off-target effect, I would say, is rather low. I mean, if I understand it, but it's a fair point here. But
Julian. A very nice talk. My name is Agam Singh. I come from National Institute of Criminology. Just for the benefit to all the audience, I would like you to briefly, very briefly explain how you reach that your MK4815 hits the falsi lysine from the starting from the parasite.
Starting from the?
Starting from the whole parasite lysate, how you reach that it is falsi lysine which is being...
Yeah. Yeah. So the first stage we did was incubate the Merck compound with the cell lysate on one hand and the live cells on the other hand. And then using CETSA we had this list with the value I showed you, the area under the curves, and then we do a ranking, and the falsi lysine came top of the ranking for these compounds, and then we check the binding, the ITC, and then...
No, my question was, how did you identify that it is the falsi lysine?
You mean genetically? You mean...
Yeah, from the total cell lysate, how did you identify that the falsi lysine is the one which is targeted?
We do mass spectrometry, yeah.
After that?
Yeah.
Okay.
Yeah.
Just for the audience.
Sorry.
Thank you.
It will be a last question. I mean,
hello, I'm from ICMR NIMR, so Shomu Chakraborty here. Yeah. So very nice talk, but a couple of questions. The Merck compound that you have shown, the binding constant is not very high actually. It's 1 micromolar. So my question is there any other protein target that this compound can bind? Second thing is, have you thought about double drugging, because it's in a separate pocket? So can you make that possible, double drugging and get higher inhibition?
Yeah, this is a very good point. Yeah, indeed, it's not the highest. So Merck compound is still a small compound, so it can be. I think the next stage is to expand to increase the interactions in the pocket, and to reach into the substrate pocket is probably too far to extend. So I think it's a good idea, but we have to grow the compound. We are growing the compound inside the pocket to increase the potency. So that's a fair point here. Yeah.
My name is Anikó Borbás. I am from Debrecen, Hungary. Congratulations for a nice talk. My question is related to the falsi lysine story. As I can understand, the Merck compound is an allosteric inhibitor, and probably it triggers a conformational change, which then leads to the closed conformation, the inactive closed conformation.
Yeah.
I think this conformational change can be further supported by molecular dynamics simulation. Have you performed such simulation to support your hypothesis?
Yeah, I mean, you summarize very well, and we did some molecular dynamics simulation, but so far even we did also normal mode calculation, but the results I've seen so far were not so. I didn't show them because they were not so convincing. But we are open for collaborations to. Now we have a set of experimental structures and we know where the hinge helix are, so we can discuss whether we can do that together. Yeah.
Thank you,
this
sir.
is the last question. I mean,
thank you so much, sir, for this wonderful talk. My name is Nirdosh from CDRI Lucknow. So my question is, as you told that Merck compound is non-competitive in nature and it is binding with the allosteric part of the protein. So, and you showed that the another compound, that is MMB, is also binding with the facility. So whether these two compounds in a competition in a computational mode will give the better result or not?
Yeah, that's a
Thank you.
good point. I think I just showed one combination of drug in the isobologram for PNP, but for falsilysine, there are some genetic studies we did with a partial knockdown, but we haven't addressed thoroughly this drug combination yet. Yeah, but it's a fair point. Yeah, thank you.
Thank you so much, Professor Julian, for such a wonderful talk. Now I request Professor Sharma to thank Professor Julian with a memento as a token of respect. I also request Professor Agam Prasad Singh to kindly thank Professor Sharma with a memento. Now we have lectures on different themes in halls A, B and C respectively as mentioned in the program schedule. Participants can attend a series of lectures based on their interest. So we will start the next session within 5 minutes.
good morning to everyone present over here. I, Dr. Maya Sharma, Organizing Committee from Faculty of Pharmacy, take this privilege to warmly welcome you all in Session 1 of 3rd International Conference of Integrative Chemistry, Biology and Translational Medicine, ICBTM 3.0. The theme of invited lecture is Biology of Infectious Disease and Drug Discovery. Now, I would like to invite Professor K.K. Dave, President, Paher University, for chair the session, and I would like to request Professor P.K. Chaudhary, Dean, Faculty of Pharmacy, to felicitate our chairperson, Professor K.K. Dave, Paher University, President. Thank you, sir. Now, I would like to request Professor P.K. Chaudhary to felicitate our esteemed speaker, Professor Yogendra Singh from Delhi School of Public Health, IOE, University of Delhi, Bharat. Now, I would like to request Professor P.K. Chaudhary to felicitate our esteemed speaker, Professor Pawan Kedhar from JNU, Delhi, Bharat. I also request to Chaudhary sir to felicitate our next speaker, Dr. Vera Alferova from Russian Academy of Science, Russia. Now, I request to P.K. Chaudhary felicitate our esteemed speaker, Dr. Soumyananda Chakraborty from ICMR, National Institute of Malaria Research, Bharat. Thank you, everyone. Now, I would like to invite Professor K.K. Dave to chair the session. Professor K.K. Dave is the President, VC of Pacific Academy of Higher Education and Research University. He also serves as VC of Indian Accounting Association Udaipur branch. He is having 26 years of working experience on senior profile as a senior manager, CEO in multinational companies, professor, director and dean in various reputed education universities. Thank you, sir.
Ladies and gentlemen, so good morning, esteemed guests, scholars and researchers. It is my privilege and honor to welcome you all to this session on the biology of infectious disease and drug discovery. Today we gather to delve into crucial discussions surrounding the intricate relationship between biology, infection and quest for novel drug discoveries. Our keynote speaker for this session need no introduction in the realm of public health and academia. We are honored to have with us Professor Yogendra Singh, the distinguished director of the Delhi School of Public Health at the University of Delhi. His expertise and groundbreaking research have significantly contributed to our understanding of infectious disease and public health policies. We eagerly anticipate this insight and wisdom that will undoubtedly enrich our discourse today. Joining Professor Singh, we have definitely would like to welcome Professor Singh to address the august audience with his specialization.
Hello, good morning everybody. Thank you Dr. Dave for the introduction and thank you Dr. Maya. Thank you very much. been working in the area of infectious diseases for almost now 40 years. And as you know now, and today this great topic where you have to understand both chemistry and biology to develop any medicine or even vaccine. So I have been working, I think I will go there, otherwise I need. Sorry about that. So, as I said, I have been working in the area of two infectious diseases. Tuberculosis, I'm sure all of you know tuberculosis is a very deadly disease. You can see, by the time I finish this lecture, 90 people will be dead. And there are many reasons for this, especially the drug resistance. And you heard the drug resistance problem, not only in tuberculosis, you heard malaria, and you talk about any other infectious disease, this problem exists. We have been studying various aspects of tuberculosis, that how does it survive? It is very amazing that we have got macrophages. I'm sure students know that macrophages are those cells which are supposed to take care of any particle dead or alive. But this bacteria manages to change the behavior of the macrophages so that it can divide and survive in the macrophages. So as I said, we have been studying many things, host response, extrapulmonary TB. You can see here, many people get infection in lung, pulmonary TB. But many people do get extrapulmonary TB. Extrapulmonary means beyond the lung. It can be brain, it can be kidney, it can be anything. And you just see the lymph node TB in the slide. We have studied microbiome, and then another disease, anthrax. Very few people know about anthrax, but this disease is important for our army. The reason is that it is a spore-eating bacteria. As you know, many bacteria make spores. The spores are very stable, and they can be used in biological war. So therefore, army of this country and many other countries, especially US, has always been interested to develop vaccine and other molecules, small molecules to treat the anthrax and even to stop the spore. So our purpose is to understand the biology of these two organisms so that we can develop the small molecule or even the vaccine. And I'm happy to tell you that we did develop the vaccine for the anthrax and given to the DRD Gwalior, a candidate vaccine. But tuberculosis, of course, is a very difficult disease where vaccine has not been working. So many people are trying. A small molecule are there are drugs, but as I said, the drug resistance poses large problem. But today I'm going to talk about post-translational modification. This is more kind of an academic, and I'm sure you know that many kinases are actually target of the drugs. In cancer, I'm sure many of you know tyrosine kinases are the target of small molecules. So, but today I'm going to discuss post-translational modification. This is a very elementary slide. Please excuse me. Only point I wanted to make is that normally we talk about these 20 amino acids, but beyond these 20 amino acids, there are many special amino acids which normally people do not read. They are diphtamide, selenocysteine, and many other. These amino acids are modified amino acids, and they are made sometime after the protein is synthesized or sometime during the protein is getting synthesized. The selenocysteine is actually made. But today, as I said, I'm going to talk mainly about the phosphorylation. This is again a very elementary slide, but just to say that one sequence of the protein can fold into only one structure. Now that structure actually decides the rate of the enzyme interaction, movement, degradation, because protein do many types of job. Now, it is very difficult to imagine. So maybe the students you should think about it. Our cells have got multiple pathways. And when you read about those pathways, you see enzymes are written into the cycles. Now, if those enzymes are there in one form or one structure, or they have only one sequence, then they always will be running at the same rate. But the pathway has to run at a different rate. The reason is, for example, when you are running, you are using a lot of energy. But when you are sleeping, probably you are using very little energy. So I'm trying to say that in the whole day, the requirement of energy actually varies with the time. Now, if there is only one sequence, then how the rate is going to change? Because in a cell, you cannot change the temperature, you cannot change the pH, and other parameters which can change to some extent the activity of the enzyme. So then what is needed then? In that case, then you need multiple variant of the enzyme which can run the reaction at a different rate. But that is also not possible because our cells are very, very crowded. This is again a question for the students. Have you thought of so many pathways running in a cell in the cytosol? How do they find their partners? How they find the substrate? How the product goes where? And how do they interact? So I think you should read about the membraneless organelles, very, very new topic. But nevertheless, as I said, the cells are very, very crowded. You can see the nucleus has got so many long DNA, many non-coding RNA, coding RNA, proteins. Similarly, cytosol has got too many things. So you can imagine if there has to be many variant of the proteins, then it will be much more crowded that cell just cannot function. So therefore, there has to be some mechanism so that the pathway's rate can be regulated. Now to explain you in another way, I'm sure people who drive manual car or even a scooter also, I guess, you have a gear. So you can change the gear so if you can have something which can change the structure of the enzyme, something you can put which can change the structure, then the rate of the protein or the rate of the enzyme can vary. That's what actually happens in the modifications. चल नहीं रहा है देखना. यहाँ भी तो कुछ आन तो है. Sorry about that. Laptop se kar do. I will say change, then you can change it. I think I should have to go a little quick. Otherwise it will be too late. So now this is for students actually it is very interesting slide. Research is okay, but understanding the biology is for students is more important. It is very funny that E. coli genome I have written the size MB, 4 point some MB. Human genome, 3000, this many MB, or you can say in GB also. So our genome is 700 times bigger. But when you consider the genes, what you see that E. coli has got 4,000 genes, and we have got about 20,000 genes. I believe all of you know about E. coli. Many of you at least will be knowing about E. coli. Do you think we are only 4 or 5 times smarter than the E. coli? So question is that how do we function like a human being, not like E. coli, even just having only 5 times more genes? So what happens when the proteins were measured, proteome was measured, what was found that there are almost 1 million proteins, although gene is only 20,000, but protein was found to be 1 million. And why? Because there was alternative promoter, alternative splicing. I'm sure you have read about alternative splicing. And then there are modifications. So actually this alternative promoters and splicing makes 10 times more protein because the different exons can combine and can make different proteins. So it becomes 10-fold more. So the 20,000 genes make actually you can say 100,000 proteins. And then the post-translational modification again increases tenfold more because there are a very large number of post-translational modifications which I show you. So that makes actually, that increases the diversity of the protein so that our metabolic pathway can run as and when whatever rate is required. Can you change the slide, please? There are many modifications, hundreds of modifications are known. Some are very less common, but phosphorylation, I have written here some major modifications. But phosphorylation actually is very common mechanism. Large number of proteins actually are getting phosphorylated, and their rate and many things actually this phosphorylation regulates cell division, movement, and you name it. Can you have the next slide, please? Just to mention that phosphorylation means addition of a phosphate group. Now, there are enzymes, they are known protein kinases, they are going to add the phosphate group from the ATP, they will hydrolyze ATP, take the phosphate, add it to the protein, and in one slide I told you that one sequence gives only one structure, but you can imagine that when a negative charge is added by the phosphate, the structure is going to change slightly, and then the rate of the enzyme is going to have different. So these kinases actually regulate multiple processes. I have written here some, and these phosphatases actually reverse the process, and eventually these two enzymes keep working on the proteins so that you can get the desired amount of energy or transcription or cell division, whatever the body is requiring. Can you have the next slide, please? I just said that ATP kinase just hydrolyzes ATP, takes the phosphate, adds to the protein, and because of this negative charge, the structure locally changes, and that, you can imagine this structure, addition of a phosphate can increase the cavity, so more substrate can go or it can close, so something like that happens, or some new sequence may open which can actually allow different interactions. Can I have the next one? So as I said, the purpose was to understand the role of the phosphorylation, and this phosphorylation actually help the bacteria also. It is in all living organism, you can say from unicellular to multicellular, including human. So I said that it's a deadly disease, kills a lot of people. Diagnosis is also very difficult. You can imagine if you have to diagnose a eye TB, you know how they do it. They will put a needle inside eye, take a sample, FNAC, fine needle aspiration, and that will be checked. So you can imagine from brain or any other tissue, if they have to check the tuberculosis infection, then they need that tissue from there. Can I have the next one? This is also, I guess, for explanation it can be. Can you just keep clicking? This is a macrophage cell. So this is the TB bacteria. Keep clicking. So this bacteria, macrophages engulf, and that phagosome is made, a vesicle is made, and that actually vesicle moves and goes and drives to the lysosome. There's an organelle where the vesicles go and fuse, and this organelle has many enzymes which actually degrade the bacteria. But as I said, that this bacteria, TB bacteria, secretes things which actually manipulate the macrophages so that this does not become acidified, and that protein is actually, I have written here, BPS 33B. So that means TB bacteria has got ability to manipulate our immune system. Can you have the next slide, please? Yeah, it degrades actually any living or next. So this TB bacteria has got 11 kinases, tyrosine phosphatases. Serine, threonine is very common amino acid which actually gets phosphorylated. I just talked about tyrosine phosphorylation, and tyrosine kinases are actually drug target for cancer in many types of cancer actually. So we have worked on many kinases, which I am not going to talk much about it because it will take large number of time. And these kinases actually regulate, I have written here, cell growth, synthesis of some molecules for the surface, for their protection. So all these kinases have got some job in TB. Can I have the next one? But what we found, this is a little old data, but what we found that Mycobacterium tuberculosis has got an enzyme which is called tyrosine phosphatase. This tyrosine phosphatase, actually, when we deleted the gene of the tyrosine phosphatase, what we found that the infectivity of the bacteria went down. But it has no problem when you grow in the culture. So what is suggested that this enzyme was not important for its normal growth, but it was meant only for us, that bacteria will secrete, this will go to the macrophages, and it will modify the macrophages so that the bacteria can manage to survive. Can I have the next slide? Similarly, we also studied another phosphatase. This was more recent data, where we found that this phosphatase actually is essential. If I knock out this gene from the Mycobacterium tuberculosis, so then bacteria is not able to survive, and you can see it causes actually multi-septation. I'm not sure whether how clear is this slide backside, but you get multi-septum into the TB bacteria. Similarly, they grow very poorly. Actually, this enzyme is required for the survival. So in the beginning, I said the purpose of this study was to understand the biology of the pathogen. Now we know that this is an essential enzyme, so one can develop small molecule which can hit these proteins, these enzymes, and eventually, if it works fine, then it eventually can be developed into some kind of a therapy or drug. Can I have the next slide, please? Similarly, we also identified that there are kinases which actually help the bacteria to modify the surface. Many pathogens have got very evil things. Actually, they, like a thief, they enter. They don't want that our immune system should be active. So what it does is actually changes the surface. So what it does actually, the bacteria has kinases, they take the signal, they actually phosphorylate a transcription factor. This transcription factor actually makes those enzymes which can add arabinase to this molecule. So it becomes lipoarabinomannan. And lipoarabinomannan does not induce our immune system. So bacteria want that, okay, in the beginning their number is less. So they didn't want to activate. So in the beginning, if the surface changes, then the immune system will not be activated and they can grow quickly. Can I have the next one? I have got a lot of data and many slides, but I'm not going to discuss more about kinases or phosphatases in TB. I will have few slides here on the anthrax part also. So anthrax is also a bacterial disease. As I said, it makes a spore. A spore can be used in terrorism, and spores are very light, so in the air it can be spread, and people who inhale can actually, if not treated in time, can die. Can you have the next slide, please? It kills humans as well as animals. It has got plasmids. Plasmids actually have a gene for toxins, and these toxins are very lethal to human beings. Can you have the next one? So, as I said, spores are very light. They can float in the air. So if I spray spore, people inhale, they can develop the disease. So people who inhale the spore, it goes. Again, macrophages, as I said, macrophages are those things which can take and kill anything from outside which enters into the body. So the macrophages take it, it germinates. Important thing is, I will explain that about in the next slide probably. Once it germinates into the vegetative form, it actually starts making toxin, and I'm not going to explain how the toxin works, but long back I showed that how this toxin is activated and how does it kill to the animal or human. So this toxin actually kills eventually. Can I have the next one? So there are a couple of proteins which actually make the whole anthrax toxin. What I wanted to tell you here was that this E. coli grows as a single cell. But this bacteria, you can see the long chains. And long chain can also be a virulence factor. That means long chain can also be responsible for killing. Can I have the next slide, please? So what was reported about a few years back, that if you inject longer chain, then it can get stuck into the pulmonary capillaries. Shorter may not, but the longer chain I think I have written here, the longer chain block the lung capillaries, causing hypoxia and lung injury. But what was not told, that what is the reason for this making chain, longer chain. So we were, as I said, we are working on the kinases and phosphatases of anthracis also. One kinase, PRKC, protein kinase C kind of a protein in anthrax. Can I have the next one? What we found, that if I delete that enzyme from the bacteria, you can see the chains becomes very short. So we gave the mechanism. So one can target PRKC and it can allow that it may become less toxic. Can I have the next slide? Similarly, this is a relatively recent data. We actually, while searching in the genome, we observed that this might be a phosphatase. We cloned and we assayed the activity. Can I have the next one? We assayed the activity, and then we thought that what this phosphatase is doing. To do that, we studied actually the morphology of the knockout strain. We seen the cell division, toxin production, sporulation, every process which this bacteria performs, we measured. Can I have the next one? What we found, that the red color, actually, if you knock out the gene, the growth becomes slow, attenuated growth. The cells become coiled. You can see they are not like a straight line, but rather they coil among themselves. They make, actually they don't make good spore. So every process of the bacteria was messed up when we knocked out this gene. Can I have the next one? And then we found out why, because there is a transcription factor, and when we knock out phosphatase, then there is a hyperphosphorylation. And hyperphosphorylated actually was not binding to the DNA, to the promoter, therefore the toxin production was less. So basically, I want to say that these kinases and phosphatases actually regulate multiple things in the bacteria, their survival, their infectivity, their pathogenesis, and so many things. Can I have the next one? This was my research part. But in human also, I have only 2 more slides, so please bear with me. So I teach MSc class, so therefore I thought just to show you that it is, for human actually this phosphorylation is very, very important, because I was talking about energy, and I'm sure you know that how the sugar enters into glycolysis and the TCA, and how the ATP is generated. So you see this enzyme, phosphofructokinase, there are 2 phosphofructokinases, and the second, PFK2, actually gets phosphorylated, and that phosphorylation actually regulates the activity of the PFK1, which is the main enzyme for in the glycolysis. So I'm sure you can read this in the book. Point I was trying to make was that phosphorylation regulates every pathway of our cells. Can I have the next one? Not only, and I have written here, you name about glycolysis, you talk about glycogen synthesis or degradation, you talk about HMP pathway, which makes NADPH, you talk about fatty acid synthesis, cell division, and you name it. Everything in the cells is regulated by phosphorylation and dephosphorylation. I think that might be a last. Do I have any more slides? This is not phosphorylation, but just to remind you that multiple modifications happen, and this is one of the modifications for your digestion actually. When you have protein digestion in the stomach, and of course in the intestine, where trypsinogen and chymotrypsinogen, so they are inactive enzymes. They are made and kept. It is not that when you eat them they are made. It is made and kept in an inactive form, but when it reaches to your gut, your stomach, then they get activated. They are cleaved, so cleavage is also a kind of a modification by which the enzyme becomes active. And as I said, there are hundreds of modifications, and each modification regulates one or the other process, and they are very, very important for survival of any organism, whether unicellular or human being. I think, so they regulate multiple processes. In bacteria, as I showed you, they regulate the survival and virulence. I will not talk about vaccine development and all those things for anthrax. So I think with this I will, can I have one more slide? So I had very large number of students. Almost more than 60 people have done PhD, and several hundred people worked in my lab as a trainee. And I was glad, and today, Women's Day, I congratulate all the women. I used women power a lot, and you can see, if you see here, Manju, Renu, Varsha, all, I will say 50% or more women were great. They were great students, and they performed very, very well. And they are all doing so well even now. Many of them are in good job in US. In NIH, they have a job in companies, and you name it. So thank you very much for your time.
Nice work done, Professor Yogendra Singh, and very simplest to understand. Now I request audience to ask a question, and we will take only one question because time is running very short, and be quick, please. Yes, sir.
How easy? So actually protein kinases also are inactive to start with. They require some kind of a signal when the body demands, you can say. So some kind of signal, then they get autophosphorylated, and then they interact. And sometimes these kinases have multiple substrates, but some are of course very specific. So for them it is very easy, because if they come together they can phosphorylate.
I mean, I just wanted to ask, like hexokinases, which are sugar kinases.
Yeah.
Is it possible that they can do the protein phosphorylation in that kind of activity?
Hexokinase, I don't think. Hexokinase might be regulated itself, its activity, but sugar phosphorylation, the activity for sugar will go down, but it will not, maybe the kinase can phosphorylate the hexokinase, and it can, with different activity, it can phosphorylate. I tried to be short. I hope I'm not too late.
Okay. Yeah. Thank you so much, sir.
Yeah.
The role of protein phosphorylations in or just starting the proteome to run the life. It was a wonderful session, and thank you so much once again. Now I would like to welcome Professor Pawan K. Dhar, Jawaharlal Nehru University, New Delhi, Bharat, on the title Dark Genome, a New Drug Discovery Platform. Professor Pawan K. Dhar leads the Synthetic Biology Group at the School of Biotechnology, Jawaharlal Nehru University, and I wish everybody, please give him a big round of applause. His knowledge talk on dark genome, a new drug discovery. Please, Sir Pawan, sir.
At the outset, I'd like to thank Professor Brijesh Rathi for having invited me here, and Dave sir for your generous introduction. Thank you very much for chairing the session. I know we are short of time, and so I'll try to speed up, and maybe during lunchtime I'll be happy to take questions if there are any. Full screen, please. Yeah. Thank you. So, well, what's dark genome? So the whole area that I'm going to describe today, which is part of the synthetic biology project, this is called dark genome, and I would like to describe our work. It has last 15 years, we are just working on only one project. Just one topic, 15 years. But it is such a huge task, huge challenge, that next 15 years also, I think this is going to be only one project. So, as you know, traditionally by converting genes into junk and garbage, we have learned functional genomics. That's the way we have grown, because you remove something, you knock down something, and you hope that you catch the expression or the function of the gene. However, the question that we asked was, can we make brand new genes? Can we cook them up real time in the lab from sequences which don't seem to be doing anything as per the annotation, the genome annotation details? So essentially, you've got three types of sequences. One is protein coding, which is minority. Bulk is RNA coding. And then some of it is left, 10%, 20%, depending on the organism, or even more, which does not seem to be doing anything. And historically, this has been labeled as junk DNA, which is not. If you look at the RNA coding fraction, that is what Susumu Ono back in 1925 called as junk DNA. Over time, we came to know that this is not junk. There is RNA coding property. However, there is still some part of it which is evolutionary, like a hand-me-down. And this is the sequence that we are talking about, the non-expressing sequences. The question is that how did nature decide where to place these start and stop signals? And there are a lot of possibilities. As you can see the sequences, there are many possibilities, a huge sequence space. How did she know that this is where I start, this is where I end? Was this random? Was this intentional? We do not know. But the question is, can we generate more start-stop combinations? Can we create our own genes in the lab, in other words? Well, if that is the claim, then one has to know what kind of sequence. Because clearly, intergenic sequences with no transcriptional history are the best candidates. And how do you know they have no transcription history? You just match them against everything that's known from the NCBI, non-redundant protein database. That's how we began. And we took 2,500 sequences in E. coli and then converted that into protein sequence equivalent, matched against the NR database. And whatever was similar, we filtered it out, removed it. What was non-similar, again, we took a small fraction. From there also, we saw what is being conserved evolutionary. We removed that also because people are going to say, okay, it's conserved evolutionary, there must be some function to this. So we don't want to take chance, we removed that. We are left with a small set, and from there we randomly selected these. There was no such publication before, so we don't know which one we should take. So the red colored line that you're seeing, arrows going back and forth, overlapping, non-overlapping, we took all the random combinations because we don't know if it was going to work. The odds against this idea were many, and when I shared this idea with my students, they refused to work on the project because they said, since there is no publication, if we fail after 2 years, who's going to be responsible? Well, we have to hire a technician from Singapore, a tissue engineering expert. We had to train her in molecular biology for 6 months, and then she is the one who delivered all the results. As a technician, she doesn't care because she gets the salary. It's not publication-based survival. So when she got the results, then everyone is interested. Then we have a huge group. The question is, how do you make a gene out of a non-expressive? Well, this is a small, very, very tiny question because you have the textbook definition of a gene, you know how it looks like. So essentially, if you theoretically put some signals upstream and downstream, as per the textbook definition, you have made artificially you have made a gene. But now if you play at the genome sequence level, it's a lot of work. You have to keep these signals in a certain order, without mutation, without any change in the distance between these elements. It's a lot of work. And we didn't have time. We are running short. So we said, okay, let's treat this as purely as information and let us pull out this information out of this genome. In other words, what we did is we created genes out of the genome where we thought that let's make a vector that supplies the promoter sequence, ribosome binding sequence, start and start and all that. It supplies constantly as a vector. So all you have to do is an undergrad experiment wherein you take the sequence, you do PCR amplification, insert in the vector, express it, there you go. So when you did that, we wanted to design a vector, and we were very, very delighted that Invitrogen had released this PVAT tope around the same time for probably a different purpose. Well, this was a divine gift for us because it cut down our work by 1.5 years at least. So now the job was just use the vector, express it. So the big moment came when we got the protein expression. In presence of inducer, there was protein. In absence of inducer, there was no protein. Where is this protein coming from? Intergenic sequence of E. coli. I want to emphasize that. This is intergenic sequence of E. coli, lab-made genes. Now, when we got this, the nature of questions changed. Of course, the question was, what are these guys doing? They're how are they looking like? So in our campus, we had, in Japan, we had 42 NMR machines. And when I gave this protein sequence to my colleagues, please solve the structure, well, they say we are so busy with Protein 3000 project in Japan. This is 5-year project. 2,500 sequence structures have to be solved by Riken scientists. The rest of the Japan, 500 structures. So there's a mega competition. 24/7, machines are busy. So I said, okay, I have to settle for computational work. So this went to my colleagues in Singapore, in the Bioinformatics Institute, who then gave me this initial data. So then the question is, what are they doing there? Are they doing some function, or it is non-functional, output? So we were pleased that, out of 10 sequences we chose, we lost 4, but the 6 ones that we had in our hand, out of them, one was, when you're expressing this gene, one was consistently killing the cell. So we said, okay, now that's a good news, because that's where the reviewers will be interested to know the phenotypic correlation of what is being expressed for the first time. And now when we did this, we repeated the experiment many times just to be sure that we are looking at the right thing. And the question was, if this is true, if there's a linear correlation between the expression and the phenotype, that is cell killing, then by switching off the gene, you should be able to restore the growth, right? And that's exactly what we found after 2 hours. When you're removing the inducer from the medium, inducer-free medium, the genes are switched off, the growth is restored back up again, thereby proving that there's a linear correlation. But the story is not so simple, because remember, we did not design the vector. We just took the commercially available vector. So in this case, the insert was not just sitting here. Of course, it was sitting here, but the stop codon was somewhere downstream. The start codon was also upstream. It was not at the edges, which means the vector itself was contributing a lot of sequences to the final protein sequence, which we did not control because we did not design the vector. As you see the red colored line, red colored alphabets, this is the actual sequence we took. Black ones are the ones that contribute to vector. So which means the previous results that you saw was a collaboration of the vector and our sequences together. It was a brand new protein, no doubt, but this is not what we designed. So we said if we have to be authentic in our claims, then we have to remove the upstream and downstream. And that's what happened. That is what happened. Okay, I think I need your help. Okay, so we used another vector and we changed the strategy. And we said, let us do the same thing but in a different way so that there is no contribution from the vector. And then we took this sequence, ECA3, which had half of the vector contribution. When we changed the strategy, it went to 0. And earlier there was no cell growth inhibition. Now cells are growing 3 times slower. And also there was some biochemical assays suggesting that it might be a DNA binding protein. Did we discover a transcription factor by accident? We do not know. There are a lot of things that we did not know at that time. So in other words, if you have to do a functional study of the lab-made genes, the only way is to overexpress them. Remember, if you're removing a non-expressing sequence from the cell, a gene knockout is okay, but an intergenic sequence knockout, no one has attempted that before. Non-expressing sequence knockout, well, you will not get funding for that because the DBT will say, okay, give me a proof that it is going to work. So the only way is to overexpress. Well, overexpression may not be the right term, maybe expressing it for the first time. And then, of course, you amplify. I think I really need your help again. Sorry. Can you change the slides for me, please? Could you change the slides? Just quick. Yeah. Okay, yeah. So the ENCODE project, as you know, it has revealed that a tiny part is proteome, bulk is transcriptome, and still there is some sequences which are non-expressing. So this is what we call as the dark genome, the non-expressing part. The question is, how many non-expressing sequences can be encoded? Now this becomes a combinatorial problem. You are starting with a set of sequences that combinatorially can evolve into millions of combinations. Now to know which combination is the one that we can work on experimentally, you need to have a database. And I think I'm really stuck. Just keep doing it manually and I'll be with you. Change it, yeah. So now you have a database, and from this database, earlier we were lucky, but we may not be lucky every time. So we said, okay, let's make a computational database. Let's profile every intergenic sequence from model organisms, assuming that this is a potential gene, and let us see what is the RNA structure, what is the protein structure, what is its function, its address in the cell, the interaction profile, and so on. Let's completely make a story out of every sequence, then we decide. Is it showing anti-cancer property? Then only make it. Which are the top 10 showing anti-cancer property? Only those sequences we pull out from the database, because we do not know if it's going to work. Next one, please. So, well, so that was the whole rationale, and we took a couple of sequences. And the first example I like to show, these are the small headlines. I'm not going deep into science because that may distract you. We produced antimicrobial peptide from the intergenic sequence of Drosophila. This was a classic example when the sequences are completely different and the structures are nearly identical. And I sent these sequences to my friend in Singapore, Chandra Verma, and he is a specialist in defensin. He has patented some molecules there. And I told him, can you look at these molecules and tell me if you can improve upon this molecule and give me a better fit. So he looked at that and he says, you can't do better, this is the best one. We said, okay, then shall I go with the experiments? All right. So we found that it is actually killing the microbes. So your experimental evidence is matching the computational prediction. So the second example is when we wanted to make anti-malarial peptides. This was done in collaboration with my colleague, Professor Shailaja Singh. So we looked into these 3 drug targets, and she reported that it is inhibiting the parasites from entering RBCs. Well, we need to repeat these results. These are some initial insights into this problem. The other idea we had is that look, tRNA for millions of years have been doing some, outreach program without any benefit to itself. It's ferrying the amino acids to ribosome, going back and forth, never cared about its own sequence. It can also be translated. After all, it is RNA. Is it possible? Well, there are steric hindrance issues. You can't do this in the cell, probably, with the current understanding. But we said, can we now think of tRNA as a sequence, computationally create a protein sequence equivalent, synthesize the protein, and now test it in the cell? So you have avoided all the problems that you would otherwise get in the cell in translating the sequence. So when we did that, we chemically synthesized a tRNA protein equivalent, and we found that it is in fact killing leishmania. We tried many different organisms. It worked in leishmania only. And as you can see, the IC50 value is very attractive, and this was then published. Likewise, Alzheimer's. This is the topic of my recent PhD student. We took intergenic sequences, and then we found that the onset of Alzheimer's, as you know, is when the plaque formation is found, and the key is really to inhibit beta secretase. So then we went through this database that we had already created, and then used some parameters to zoom into the sequence that we would experimentally create, and then see the effect. And the outcome of this approach was that, as you can see, we achieved 95% inhibition of beta secretase. This was very encouraging. Now we are trying to do some animal work and looking for collaborations. We run out of the money and hoping that someone will help us in doing the experiments. Some of the computational predictions that are undergoing experimental validations, we are talking of now antisense protein. That's one of my students who is here, Mohit. He is trying to repurpose the drug genome. You have a sense strand, you have an antisense strand. In the antisense strand, we have done a lot of bioinformatics, and we have chosen our sequences. These are experimentally being produced, and we will know the results hopefully in a month or 2. Likewise, I think I need again your help. please change the slides? So the second one, next one. Yeah. Oh, I think we just skipped one. All right. Can you go back? Yeah. So we are also doing the, no, I think maybe back? No, pehle le jana. हाँ जी, yeah. So reverse protein. What is reverse protein? You have the sequence, as you know, it expresses in a certain direction. So we said, can we get a full length translate of the same sequence, protein coding sequence, in a reverse manner? When you express in a reverse manner, you have a new protein, and a brand new protein that nature probably didn't look at in that organism. So we are now, I think, at the verge of producing some results. So hopefully in a couple of weeks we should have the first results of making reverse proteins. Intronic proteins, likewise, philosophy is the same, vector is the same. It is just that we want to see how far we can stretch our imagination beyond which this idea will not work. Likewise, non-coding RNA being translated into proteins. It's a new biology. We do not know how many failures we are going to produce, we're going to come across. Nevertheless, this is an exciting space. Why it is exciting? Because it's an untapped gold mine. People are looking at protein coding, RNA coding, but there's a lot of stuff in the non-expressing genome. And what we are presenting today is some of the examples of a new drug discovery platform. The best part is you own the IP of every molecule that you make, because you do not have the evidence of that in the nature, so you can name it after your name. Sorry, next one please. So the dark genome word has been used in different contexts. People are using, however, our definition is very clear. And this is also our boundary condition, that it is non-expressing, non-translating, and extinct genes that are artificially encoded into functional molecules. So I try to capture our journey, 14 years journey, in the form of this small book. I could make it below rupees 78 because that's what Amazon has a limit. We wanted to make it free, but then that's the best we could do. So if you're interested in this area, please have a look at this work, and then hope. Well, this paper got published in 2009, and European Science Commission picked up this work, and then after the talk, they gave us some interesting, encouraging feedback, and then it kept on going, and industry is now looking at it a little more seriously. Imagine you're artificially expressing 98.5% of human genome into functional peptides and proteins. That is the scale. There is so much out there that just one lab or maybe 5 or 10 labs are not enough to do this work in the next 10 years. So we are looking for collaborations. We're looking at the evolution of functional genomics into 2.0. And we are now, we have enough confidence after 1.5 decades that it's time to launch a consortium. So we are working seriously on that. And hopefully you will have something good next time when we meet. Everything started from junk DNA, junk genes, RNA proteins, and so on. So sometimes my students like to call this area junkomics, a new area of doing biology. So these are the people from Singapore and from Riken, the group there, and Professor Sakaki. I must be very, very highly thankful and obliged to Professor Sakaki, who was the president of Human Genome Organization at one time. When I joined Riken, he told me, what do you need? I said, funding. Okay, give it. No project was submitted, no review. $3 million was given. Do what you want to do. No questions asked. If I had submitted the project to DBT, it would have been rejected. So this is the way the science is done overseas. They trust you. Whether you fail or succeed doesn't matter. But at least you're making a sincere attempt. And after we produced the initial results, we were encouraged to go further, and now we're talking with some industries to realize its potential further. So I think I have a last slide of this. Yeah. All right, so that's our small university. And if you wish to know more about it, that's the website. So thank you very much for your kind attention.
Thank you.
Yeah, we have room for one question, sir. Yeah. One question, yeah, please.
Hello. Thank you so much, Pawan sir. Now we would like to take one question. Yes, please. Yeah, just wait. Just wait.
Hello.
Yeah, please.
Yes, you can ask.
First of all, that was a very wonderful and, to be very honest, a fascinating lecture I've ever heard. My name is Shri Babu and I'm from Hansraj College pursuing my zoology BSc. My question was regarding that. What are the biochemical mechanisms underlying the evolutionary conservations of dark matter regions across species?
We don't have enough data, honestly, to answer your question. First of all, the dark matter definition itself needs to be settled, because some people are calling unannotated areas as dark matter. Right. For us, that's not the dark matter. We are given very crisp 3-parameter definition within which we want to stay. To answer that question, I can give a broad answer. I don't have the data to back up this answer, but I would say that different organisms are using different proportion of sequences. Now, whether it is intentional or it is accidental, we do not know. Right. Conservation of this is not there, because that's our filter in the first place. Whatever is conserved, we don't call it a dark matter, because it will have some role somewhere. That's why the nature has preserved it.
Right.
So, so our parameters are very clear, but I think the question has to be evolved into a different setting, which means how much of human genome is human? Very little, you'll be surprised to know. This is all hand-me-downs from bacteria. 99% of our genes are very similar to that of mice.
Right.
Okay, I'm sorry, you want to say something else?
No, sir.
Okay.
That was my basic question. Thank you very much.
Okay, thank you.
Thank you, Pawan sir. One topic, 15 years, one project, dark genome. Still 15 years more you can. So there is a large scope, and your dedications how to get research is very difficult to be, but integrity is your highest, and that we can learn from him itself. So thank you so much once again. Nice interaction. Thank you so much. Now I would like to call upon Dr. Vera Alferova, Russian Academy of Science, Russia. Her title is on current trends in natural antibiotic discovery. Dr. Vera Alferova serves as a senior research fellow in the Laboratory of Microbial Resistance within the Peptide Protein Technology Department at the Shemykin Ochinkov Institute of Bioorganic Chemistry, Russia Academy of Science. Now I would like to welcome her, and please give her a big round of applause.
Just go next. Just go previous.
Is it working here?
Yes, working.
Good morning, colleagues. First of all, I would like to thank the organizers of this event and Professor Ratti for his kind invitation for me to participate in the event. I work in Institute of Now it's better. Okay. I work in Institute of Bioorganic Chemistry, and for the past few years my work was concerned with natural products, and I would like to tell you about the current trends in natural antibiotic discovery. First of all, about what antibiotics are as a term. This term was first defined by Zellner Waxman in the middle of 20th century as a chemical substance of microbial origin that could inhibit growth or even kill microorganisms. But since then, this term was widely used and transformed multiple times, and there is no strict definition of the term nowadays. In narrow sense, antibiotics are small molecule antibacterial drugs used in clinic or veterinary medicine and produced by microorganisms. But actually, in literature, one can find that every of these criteria is not essential. So we can find the mentions of antibiotic peptides, antifungal antibiotics, anti-cancer antibiotics, semi-synthetic, synthetic antibiotics. So this term is quite broad, and just taking this into account, I will further discuss the specific antibacterial small molecules as antibiotics. Antibiotics played a very important role in the increase of life expectancy in 20th century. They decreased dramatically the mortality from infectious diseases, wounds, sepsis, and surgeries. But nowadays, we are facing the antibiotic-resistant problem. There are many variants of this slide for this illustration, how pressing this issue is. Here is the information from the review that was published a little bit more than a year ago, and it summarizes the information on the frequency of isolation of resistant isolates around the globe till 2019. It's interesting that this review was published only in 2022. And during 1 year since its publication, it was cited more than 5,000 times. This highlights the importance of this topic and the high interest of the academic researchers to this problem. So, if this issue is so pressing and interest is so high, why there is so low rate of new antibiotic introduction? I would say that one of the problems with this is lying not in the academic researches on this topic, because antibiotic research and development is almost has the same cost as other drugs, but and they have the same success rate, but the expected revenues for the manufacturers is significantly lower. First of all, the resistance is mostly nowadays problem of low-income countries. Then the most effective newly introduced drugs that are effective against resistant strains are mostly held in reserve. So they are used only in the most extreme cases, and therefore they are not produced in large quantities. Taking this and some other reasons together, the expected revenues for the manufacturers is very low, and therefore pharmaceutical companies are mostly not interested in antibiotics as a product. And in our country, we have very, very low or non-existent governmental support for introduction of our novelly discovered antibiotics in the clinics. So we mostly have to strict just to the academic research. But as for the academic point of view, natural products are still very interesting as the sources of new drugs. Here on this slide there is a diagram of the origin of all drugs introduced into clinical practice for 40 years. And we can see that natural products unaltered, just as they were found, make up a small share, about 5%. But only 1/3 of these compounds are completely synthetic, and 2/3 have somehow natural origin. They are either derivatives of natural products or synthetic compounds, but based on natural scaffolds and pharmacophores. This makes studies on natural bioactive compounds very promising as a source for drug leads. Microorganisms are ubiquitous in nature, and in the so-called golden age of antibiotic discovery in the middle of the 20th century, we could study almost every organism alike, and we had a significant probability to find something new with a broad spectrum of activity and clinically relevant. But nowadays we are facing a rediscovery problem. And if we just study microorganisms we find easily, we mostly discover the same compounds again. But in nature there are some ecological niches that are more interesting because the evolutionary pressure there tends to a higher possibility of clinically relevant features of secondary metabolites. For example, microorganisms associated with the animals are more likely to produce the compounds with low toxicity to mammals. Actually, facing this rediscovery problem, we almost always find something not interesting for us, and all our efforts to make this percent of interesting objects higher, including target-based screening and mechanism-informed approaches, our